So today I want to talk about the taste and smell physiology. As you see in the picture, there are some receptors in here that today in this first video I will talk about the taste, and in the next video I will talk about the smell. All in all, the receptors which respond to the molecules that are dissolved in fluid are chemoreceptors. The chemoreceptors can be for taste, gustatory receptors, or can be for smell, olfactory receptor. The taste, it means that the molecules will come and dissolve in the fluid of the mouth, salivatory fluid of the mouth. And for the smell, the molecules are going to be dissolved in the mucosa of the nose, or in the for the taste, in the mucosa of the mouth. So in here, as you see, I want to talk how it happens. The molecule will come to the taste pore. It will enter to the taste pore. It will jump into the taste pore. And there are some other cells in here. These purple cells are gustatory taste cells. They are the epithelial cells which are changed in a way that they have been specialized, and they are called the gustatory taste cells, and they are the epithelial cells, but they are the specialized epithelial cells. And in here we have a neuron in here, which is a sensory nerve. This is the secondary sensory nerve, because in here this epithelial cell, which is a specialized cell, which has changed, and right now we call it the gustatory cell. This will finally, because of the taste, for example, the molecule comes in here, for example, the sugar, or for example, the salt comes in here, and these molecules will combine, and finally the neurotransmitter releases from this gustatory taste cell. So this is the secondary sensory neuron. And in here there are some other cells. They are the supporting cells in here. Between each gustatory taste cell there is a supporting cell, and this completely is called the taste bud. There are lots of taste buds, as you see in the picture. This is a small part from the tongue, and there are lots of taste buds. And also there are 50 till 100 epithelial taste cells in each taste bud. And in here, as I told, this is a taste pore. What happens? The taste buds in the anterior 2/3 of the tongue are innervated by the facial nerve. The anterior 2/3 is innervated by the facial nerve, which is the 7th cranial nerve, and in the posterior 1/3 is innervated by the glossopharyngeal nerve, or cranial nerve number 9. So anterior 2/3 for the facial, and the rest is for the glossopharyngeal nerve. And the dendrite endings of the facial nerve are located around the taste bud, and they are responsible for the touch and also temperature. So the facial nerves are responsible for touch, temperature, and also tastes. But glossopharyngeal is only taste, and it is the posterior 1/3 of the tongue. So what is the mechanism? First of all, before the mechanism, there are major categories of taste in the old physiology and also in the modern physiology. The old physiology says that here is for bitter taste, here is for sour, here is for salt, for sweet. It means that each part is responsible for one taste. But the modern physiology tells that, first of all, there is a 5th category of taste, which is umami, which is tasty. It is the taste of being tasty, but related to meaty flavor. And it is for the amino acid glutamate. And the flavor enhancer is N-glutamate. And another part, which is for the new physiology, it says that bitter, salty, sweet, and umami and sour all can be felt in any part of the tongue, but the density of these receptors is different. And in here, there is a mechanism for the taste. If it is the salt, so salt has NaCl, so it has Na and also Cl. And Na is responsible for this mechanism, which I want to tell. Na is an ion, and it is small. So in here, there is a receptor. It will come inside the cell. It is Na plus. It means that it causes so many positive ions in here, and finally depolarization happens. Depolarization happens, and finally, as the depolarization is happening because of the positive ions in here, the calcium channels, which are the ion calcium channels, the voltage regulated calcium channels, will be open too, and the calcium from outside will come inside the cell, and finally depolarization will be a lot, and the receptor potential happens in here. Finally, the neurotransmitter is released. This neurotransmitter will come and join another receptor, which is on the membrane of this secondary sensory neuron. So the neurotransmitter will join the receptor. The receptor will open another channel, which these are the ligand operator channels, and then the positive ions will come inside this cell, In here, we have a generator potential, or EPSP, excitatory postsynaptic potential, and this excitatory postsynaptic potential, or IP or EPSP, or this generator potential, which is generated in here, will come along this sensory neuron, which is the secondary sensory neuron. If there will be a voltage regulated sodium channel, which is regulated because of its voltage, exists in here, it will be open and lots of sodium will come inside the cell. The depolarization is high and it has reached the threshold level, so action potential happens, and the taste sense is going to the brain and tell the brain, okay, here was salty. So this was about salty. For the SOR, we have the same mechanism, because this H plus, which is responsible for the taste of SOR, is small enough so it can enter the cell from this receptor. It can enter the cell. In here, depolarization happens, and calcium channels are opened again because the depolarization is happening. So there will be lots of positive ions in here. Finally, the neurotransmitter is released. And the receptor potential had happened before that, not action potential, not EPSP. The receptor potential had happened, or the graded potential. In here, neurotransmitter is released. It will connect to another receptor, and the channels will be opened. As the channels are opened, then it will bring the information. As the channels are opened, then the ions, which are the positive ions, will come inside the cell, and it will bring the information. As the EPSP, it will bring the impulse, the EPSP, the excitatory postsynaptic potential, which has happened in here, it will bring it throughout the secondary sensory neuron. And if there will be sodium voltage regulated gated channel in here, the sodium voltage regulated gated channel will be opened, then the sodium ions will come into the nerve fiber, and final action potential happens. And this action potential is a stimuli. It has happened because of the stimuli, and will go to the brain, and then the brain understands that, okay, here we had a taste, which was SOR. So for the sweet and also the bitter, the story is a little different. And why? Because sweet, the molecules which are related for the sweet, which is the glucose, fructose, lactose, sucrose, and every sweet molecule else, they are big, so they cannot enter the cell. So there must be a receptor in here. It means that they are the ligand operator receptors. And a ligand, which is a molecule, which is, for example, C6H12O6, which is the glucose, for example, or fructose, or whatever, any molecule which is responsible for the sweet taste, will come and attach this receptor in here. Then the G protein will be activated. This G protein is consists of alpha, beta, and also gamma parts. This G protein is called gustocin. There are several G proteins. One of them is gustocin, which is responsible for gustatory taste. So it is logical. The name is gustocin. In here, the G protein will be activated. The activated G protein, which is the alpha subunit, will come and activate adenylate cyclase. Adenylate cyclase is an enzyme. Adenylate cyclase, ACE, it has ACE, so it is an enzyme. And this enzyme will come and change the CAMP, or the second messenger, which is CAMP. The cyclic AMP, it will the adenylate cyclase will come and make the second messenger. This second messenger is called the CAMP. This second messenger will come and close potassium channels. So in here, the polarization finally starts. Why? Because these potassium channels before that would let the potassium goes up, the positive ion goes up, but right now it will save the positive ions. So in here, depolarization happens. Because of this depolarization, the neurotransmitter, which had been made before, will be released in this synaptic area, in this synapse, and this neurotransmitter will come and join another receptor. This neurotransmitter will not enter the cell. It will connect to the receptor, and then the channel, another channel, which is the ligand operated channel, will be opened, and then the sodium ions and the positive ions will enter the cell, and EPSP is made in here. This EPSP, the summation happens, the temporal summation, spatial summation happens in here if it is strong enough, if it didn't die out because of, for example, sodium potassium pump, which will bring 3 sodium out, 2 potassium inside. So it will regulate. It is in fact fighting with this EPSP. It is the opposite part. So if this EPSP will be saved and the positive ions in here will be a lot and enough, which they reach this potential, which is their generator potential, or EPSP, will reach the sodium voltage regulated gated channels, and they are enough. So this channel will be opened, then a lot of sodium will enter the cell. The action potential will be made because, in fact, depolarization has reached the threshold level and will go to the brain finally. So the point was this, that the G protein is activated, the adenylate cyclase is activated, so the second messenger, which is the CAMP, is made, and this is caused, so the channels of the potassium will be closed. For the bitter taste, we have another story again. We have the molecule name is quinine. The molecule is coming in here, attaching to the receptor. The G protein, which was the gustocin, will be activated. It has three parts, alpha, beta, and also gamma. The alpha part is the activated part. It will activate the adenylate cyclase, which is an enzyme, and it will make a second messenger. The second messenger for the sugar was CAMP, but in here the second messenger is different. Finally, the second messenger is caused that the calcium is released from the inner part of the cell, not outside. For example, in here, the calcium was from outside for the SOR, for the salt, the calcium was from outside, but in here, from the endoplasmic reticulum, it is released, and the neurotransmitter finally is released because depolarization has happened, receptor potential has happened. So in here, the graded potential has happened, so neurotransmitter in here is released, and finally, and touching the receptor and attaching to its own receptor, and the neurotransmitter in all these parts is glutamate. And finally, the generator potential, or the EPSP, if it is strong enough, if it didn't die out, if it could handle this situation, it means that if it could fight better according to the contraction rule to the sodium potassium pump, then it will come and attach to the then the stimulation will come and continue, and then finally we have a sodium voltage regulated gated channel. They will be open, the depolarization is so high it will reach the threshold level, and finally action potential happens and it will go to the brain. There are some other notes about these tastes which I want to mention. First of all, for the salt, the onion modifies the perceived saltiness. It means this coloring here, which is a negative ion, which is onion, it modifies. It means that the perceive and the perception of this Na and also the salt is better when the color exists, and exactly when it is colored. Another role for this color is this, that these taste receptors, these cells which are responsible for the taste, they are attached together by the claudins, by the proteins which are called claudin. They are responsible for the tight junction between the cells. This color is going through these tight junctions because this color is small enough. It can pass through the tight junction, and then it can go out of this area. Another point about the sweet taste is this, that the sweet taste of the amino acids like phenylalanine or tryptophan, and also the artificial sweeteners like saccharin or cyclamate may enlist different second messenger system. For example, the inositol triphosphate or the acetylglycerol. In here, the second messenger was cyclo-AMP, but for the artificial ones, for the amino acids which are like sweet taste, like for example phenylalanine or tryptophan, or the artificial sweeteners like saccharin or cyclamate, the second messenger is different. It is inositol triphosphate or the acetylglycerol. And in here we have the gustatory pathway. The gustatory pathway tells that we had 2/3 of the anterior part of the tongue was because of the, the seventh cranial nerve, the facial nerve. The posterior 1/3 is the ninth nerve, which is the glossopharyngeal nerve. They are picking up the sensation according to the tastes, but the facial nerve can have another branch and can pick up the pain and also the temperature also. So the facial nerve has two branches from the 2/3 part of the tongue and also from the palate. And also in here, the throat is having some senses by the 10th cranial nerve, and they are all picking up the sensory information to the nucleus tractus salitorius, and from the nucleus tractus salitorius, the information is going up. The nucleus tractus salitorius is located in the medulla oblongata in the brainstem, but finally the information is going up to the hypothalamus, to the thalamus, and also to the limbic system. What I wanted to tell is this, that in the limbic system, hypothalamus, and also in the thalamus, they will have some branches, and finally it will go to the post-central gyrus and also the insula. In the cortex, in the post-central gyrus and in the insula, it is the conscious part, it is the part which analyzes, but the limbic system, which from the medulla oblongata, from the nucleus tractus salitorius, the nerves are coming to the nucleus tractus salitorius, which is located in the medulla oblongata, and finally it will have some branches in the limbic system. And why limbic system? Limbic system is connected with our emotions. It means that I will tell, for example, in here there are some emotions synapses, there are some synapses which are related to the emotions, and I will say, okay, I don't like this taste, but I don't know why, or I like this taste, but I don't know why. This is emotion, and it is related to the limbic system and also the hypothalamus part. But the thalamus, finally, by the tertiary nerves, it will go to the postcentral gyrus and also the insula. That's it. So this was all about the taste, and in the next video I'm going to talk about the smell.